Ray, go ahead and take it away.
Okay, thanks, Bruce. Hello, everyone. My name's Ray Wheeler. I'm a plant physiologist at NASA's Kennedy Space Center. And today, we'll try to address some plant physiological challenges that you will face to conduct space flight experiments and space exploration experiments at other settings with plants. And then we're doing sort of a tag team approach with this. I'll go first, and then my colleague Gioia Massa will go second and focus more on the production of food with plants. So I've broken out my approach in terms of the challenges, the plant physiological challenges, into several different categories. And others might label these different, but some I'm calling direct gravitational effects in the space environment. For example, if you're in low Earth orbit or a transit mission, you're essentially weightless or you're in microgravity. Then there are some indirect effects from these gravitational environments, and I'll define that a little better when I get to that. Then there's some just general effects of the spaceflight environment. Being inside a spacecraft, there are other things that are happening there that can affect plants and their physiological responses. And then, as I mentioned, Gioia will follow up with this on crop production challenges. So, okay. So there's some constraints that we all face when you're trying to grow plants in space. And this is irrespective of the setting or destination. You're usually always constrained to small volumes. We don't have large greenhouses or very large plant growth chambers. So that you need to consider that in terms of plant research and plant production systems for space. We're also usually typically limited in the time we have to conduct these experiments. And so you have to consider that in terms of your experimental design and how you approach things. Power is almost always limiting, and that'll be more of an issue for production systems. You're also limited by mass, and then the amount of crew time that you need to support these experiments. So crew time is a pretty precious commodity depending on the mission, and so if it's pretty intensive in terms of requiring crew time, that's going to be a challenge to get that much available to you. Now, the life support community has combined all these kinds of constraints into a more unified metric they call equivalent system mass. We don't need to dwell on that much, but as you begin to think in future systems of if you do produce plants for food and other life support commodities, how do you compare these with other technologies? And this equivalent system mass or ESM metric is one way that's done. That's just sort of a side comment. Hopefully I can advance. There we go. Okay, so direct gravitational effects. Plants in satellites or that were flown in the space shuttle or in orbiting space stations like Mir and the ISS are in a perpetual state of free fall. So we refer to that as microgravity. So how do the shoots and roots and other organs orient and grow? Just a fundamental physiological response to the environment. How does microgravity affect things like tropisms and nastic responses or mutations? And just to kind of show you some real world examples of plant organs that are showing strong gravitational responses, here are some pictures. You see a Norfolk Island pine on the left there that's showing a strong negative gravitropic response in terms of the main trunk of the tree, while the lateral branches are showing what's referred to as a diagravitropic response, almost 90 degrees to the plumb line. So it depends on the organs. There's some shoot organs that are even positively gravitropic. If you see the center photo at the top, those are peanut gynophores. Those are the elongating components of the pollinated peanut flour, and then they go straight down the plumb line. So it's a shoot organ that's positively gravitropic. Another example of that, to the right is a picture of a banana fruit cluster. You'll notice that the main cluster itself is positively gravitropic, and that bottom part of the banana inflorescence is called the bell. That contains a lot of male flowers that continue to flower, whereas the fruits, as they grow, develop in a negatively gravitropic fashion. And then there's some other organs, even subterranean organs, that are going at angles to gravity. In the center bottom, I show a picture of potatoes that were grown in an aeroponic system, and you see the stolons, the underground stems or rhizomes, that are going at an angle to the gravitational plumb line. So these are all interesting phenomena that you might consider in terms of direct gravitational responses to plants. One of the original interests, and I won't dwell on this too long, I show a lot of pictures here, but one of the original interests of sending plants into space was, will leaves become epinastic? Because this had been observed for many years in clinostat studies. It goes back 100 years. And so an experiment was designed for Biosatellite 2. This was one of the first plant experiments ever flown by the US. And the pepper leaves did indeed become epinastic. And epinasty is a downward curling of the leaves. And they did it in space, and they also did it on ground-based clinostats as controls. So that was a really neat observation. And I don't know that this is still a fully resolved thing for space in terms of microgravity. Do leaves initially become epinastic, and do they then acclimate and grow more normally? This is an interesting thing that I think needs further testing. It was kind of a fun study, and I thought from a historical perspective, it would be nice to mention. Because of this, even very early in the space program, there was a strong case to be made by a lot of the investigators to have centrifugation capabilities in space. And the reason for this was, if you wanted to do 1G controls, you could do those right side by side with the plants that were exposed to microgravity. And I show some examples here of centrifuges. On the very left is what's called the gravitational threshold plant gravitational threshold response facility, and it had two centrifuges in it. And at the bottom there, you see Dave Chapman. He was at the University of Pennsylvania and helped build that hardware in the 1980s, and then he moved to Kennedy Space Center. A more recent example of a centrifuge facility is the EMCS, the European Modular Cultivation System, shown on the right there. That was on the International Space Station for many years. It has since recently been taken down. But so centrifugation capabilities, and that allowed, say, for example, the University of Pennsylvania group to actually study things like circumnutation of sunflower seedlings side by side in space, both at 1G and at essentially 0G. Another example of some experiments done on centrifuges, this is a photo courtesy of John Kiss from some of his research on the space station. In a lot of his studies, he validated that blue light is a strong orienting clue for phototropism in plants. This was a well-known phenomenon in plant physiology, and space is a great place to study phototropism. But he also, and I think this is really neat, when he exposed the plants, the germinating seedlings, to unilateral red light, he also saw a positive response. And so this was something that wasn't really even known before John conducted these experiments in space flight. So just to give you an example of how effective a free-fall microgravity environment can be to conduct things like phototropic studies. I just show you some other neat examples of interesting phenomena. These are moss protonemata cells is the proper plural. This was work done by Volker Kern and Fred Sack at Ohio State University. And what they noticed is that in microgravity, the moss protonemata would kind of grow in a spiraling fashion, in a clockwise fashion. And other related phenomena like roots sort of deflecting or skewing in one direction has been observed by groups, Annalisa Paul's and Rob Furl's group at the University of Florida, and likewise John Kiss's group. So some interesting phenomena that occur in these odd settings of microgravity. So now, indirect effects of microgravity. When you don't have any gravity, you don't have any natural thermal buoyancy or any natural thermal stirring of the air. So if you have an enclosed system where there's no active convective air movement, you'll get a very still environment. And as an example of that, the early plant growth unit used by NASA, I showed to the right there the plant growth facility, which was quite similar, but was the plant growth unit essentially had closed chambers for plants, and there was no natural stirring of the air. And so the observations from this, they saw unique observations depending on the studies, but as they began to think back on them, they weren't quite sure, is it really a direct gravitational effect on the plants, or was it a secondary effect from lack of poor air movement? There are also a lot of concerns in terms of water and managing root zones in a weightless environment, and even in fractional gravity environments. And Gioia will expand on some of this. Scott Jones's group at Utah State University has done a lot of interesting research along these lines, and a lot of interesting modeling work. But as a consequence of poor drainage, you could expose your plants to root zone hypoxia. And so this is a consideration for spaceflight environments and conducting good plant growth experiments. Just as an example of this, a colleague, Lucy Poulet here at the Kennedy Space Center, where Gioia and I work, allowed me to show this graph from a paper she published in Astrobiology. And what it shows is, on the x-axis, the acceleration due to gravity. So if you're in microgravity, you're right there at the origin at 0. And then she modeled different air speeds and predicted the boundary layer. So that's the still layer above the leaves that would occur in these different combinations of gravity and air circulation. And what you'll see, that top line where there is 0 air movement, 0 meters per second of airflow, when you get to a 0G or very low gravity environment, you have these very thick boundary layers, which is not good if you want good gas exchange to the plant leaves. So speaking of gas exchange, there have been plant growth chambers designed that could accommodate those types of measurements to allow measuring things like net photosynthesis, respiration in dark periods, and transpiration, throughout growth cycle. And I show some examples of these. It goes all the way back to a very early experiment conducted by Herb Ward on an Air Force satellite with duckweed. Then more sophisticated chambers like the astroculture chambers you see in the lower left, the biomass production system, which was essentially evolved from the astroculture technologies. And then to the bottom right, probably the most sophisticated plant chamber that has these kinds of capabilities is the advanced plant habitat that NASA built with Orbitech in Sierra Nevada. And you see that it can be totally enclosed, allowing direct measurements of atmospheric gas exchange. So that's a very handy tool to have if you have the right kind of chamber in space. So other effects of the spaceflight environment. Not only are you facing different gravitational environments, you're facing other kinds of influences from space settings. For example, if you're open to a space cabin environment, there are volatile organics that are sort of blowing around. The carbon dioxide can be very high, even to almost super elevated levels. Depending on the mission, this isn't the case for the ISS, but future missions might be at reduced pressures. So that's a consideration that you need to include. And of course, as you get into space, depending where you are, you're going to have different radiation environments. That's not really my area of expertise, but this is clearly an important area of in terms of the environmental influences on plants and all living things as you get, especially when you get beyond the protective magnetic fields of the Earth. Some examples of trace contaminants, trace volatiles that can build up. There are all kinds of things. Clearly, the one that's high on the list for plant chambers is ethylene gas. Plants make ethylene, they use it as a hormone and as a growth regulating compound. And so that's one you want to watch very carefully in any kind of closed environment. It could be in the spacecraft itself, it could build up in the plant chamber. So you need to be aware of these kinds of organic compounds that can build up in closed environments. I mentioned the CO2 can also be high in spacecraft. This is a plot of the CO2 concentrations on the International Space Station up until about, I think it's 2012 or so, on the right side there. And you'll see that the CO2 ranged anywhere from, say, 3,000 to 6,000 parts per million. So that's about 10 times the natural ambient CO2 concentration on Earth. Now they've made a concerted effort to bring that down. It's more like 1,500 to 2,000. But nonetheless, the point being, if you're open to a cabin environment, your plants would also might be exposed to these very high CO2 levels. And just as an example of what can happen, and this was unexpected, if you do expose plants to very high CO2 levels, in this case 10,000 parts per million, this shows a measure of stomatal conductance. These were potato plants through a light-dark cycle. So conductance plotted on the y-axis, and then the time of day on the x-axis. And if you look at the 400 parts per million, 400 micromoles per mole, you see a very typical curve. The stomata are closed, partially closed at night. They open up light cycle and then begin to close down again as darkness approaches it. So it's a circadian rhythm and trained type cycle. When you raise the CO2 to 1,000, you reduce that opening. And so this is a well-known phenomenon, but if you get to super elevated levels, like 10,000 parts per million, the conductance actually goes up compared to ambient, and you don't get much closure at night. So you tend to damp out the circadian rhythm of the stomata. So I just mentioned this to show that there can be some unexpected or peculiar phenomena that occur in some of these extreme levels. Pressures could be different for future missions. They were different in the past. Skylab and the Apollo missions were all at 1/3 of an atmosphere, different oxygen partial pressures. The shuttle and the ISS are at 1 full atmosphere and about 21% oxygen. Likewise, Mir, but maybe that won't be the case. And there's a lot of discussion for future habitats and lunar settings and at the Gateway that you'll hear about in other seminars in this series talking about these settings. So pressure is another phenomenon to consider. If you do have reduced or different pressures, you have to think about things like gas diffusion. That's a direct function of the external pressure around, say, a leaf. And this just shows an example of that. As you go down in pressure, the diffusion coefficient of all gases goes up. In this case, this is a model for carbon dioxide taken from a paper by Corey et al. But it's a function of Fick's law of molecular diffusion. So this will affect things like transpiration and gas exchange with plants. Plants grown in space and plants that will be grown in the future will probably be grown under electric lighting systems. And the clear winner in this approach right now is LEDs. They have just reached remarkable efficiencies, and Gioia will expand on this. So I won't dwell too long on this, but LEDs are narrowband sources, so you have to think about how you would compose different combinations of LEDs. how does this affect the plant physiology directly through the lighting environment. But they become a fantastic tool for growing plants in controlled environments. And by the way, the use of LEDs to grow plants was patented through NASA-funded technology to the University of Wisconsin in 1990. And this just shows that might not mean much to you, 3 micromoles per joule, but take my word for it, that's just a remarkable efficiency for an electric light source. That's also some recent NASA-related work from Bruce Bugby's lab at Utah State. I won't talk too much about this. Joy will get into this, but depending on the gravity environment, the amount of water drainage and where you get perched water tables also will be affected. And I mentioned Scott Jones's lab at Utah State has done a lot of modeling on that, and just checking my time here, Bruce, let me know. So you have to think about this. Depending on the setting and the gravity, this is going to affect the water drainage in your, if you have a solid media, say a potting media. And there's some other issues that you have to think about when you get into space as well for in terms of evaporation. I won't say much about this because I know Joy is going to address that. So I'm going to end there, and then I will try to pass the baton to Gioia. Oops, sorry, wrong control.
So thanks
for back there.
I think Gioia, you should be able to just share your slides now, right? And that should take it over from Ray. Gioia, you're still on mute.
I'm trying, but it says can't display content for some reason. Can you stop sharing, Ray? Let me see if that helps.
Let me see if I can.
If you go up next to your microphone, there should be a box with an X, perhaps.
Yeah, it's between the microphone and leave. Just click on that.
Okay.
For some reason, it doesn't. Let's see.
Turn the microphones. Okay. Stop sharing. Got it.
Okay. Let's try. Well, I'm going to do window. Let's see. Okay. Can you see my slides now?
Yes, we can. Thanks, Joy.
Okay, great. So thank you, Ray. So I'm going to talk a little bit about the specific challenges we're thinking of for crop production in space. And so when we look at characterizing these, we can characterize them, by where they are. So we can kind of think of some specifically for deep space, things like microgravity, and the fluid behavior and how fluids behave so strangely in microgravity. With the lack of the convection that Ray was just talking about. We can think of surface-specific challenges, things like dust or partial gravity. And then there are some challenges that are kind of shared with both of these environments, radiation, micrometeorites, the reduced pressure that Ray discussed. There are some challenges that are really specific to crops. Thinking about productivity, stress tolerance. These are the same challenges that we have in terrestrial agriculture, environmental optimization for indoor agriculture, crop scheduling. But then some which are common to all of these. And so those would include things like, the sustainability. How do we become more sustainable when we have these limited vehicle resources and limited crew time? How do we deal with these abiotic stresses? And what is this closed environment microbiome going to be like? And how can we make it work for us? So the ones in red, fluid movement, radiation, high CO2, microbiome, these are things that we're actually working quite a lot on right now at Kennedy Space Center. But we've identified a whole list of gaps and technology and knowledge gaps. And a few of the higher ones are kind of highlighted here. And these are, I'm not going to go through each of these because I will go through them in the talk, but these are some of our big heavy hitting gaps. And when we look at the research that we're doing on the International Space Station with the Veggie chamber, we actually are addressing quite a few of these. So all of the ones in green are being addressed with Veggie. So I'm just going to go through a few of these and talk about how we're working on these right now with Veggie and what we're thinking about for the future. So Veggie, for those of you who aren't as familiar with it, is a small plant growth chamber that we have on the International Space Station. And here you can see astronaut Shane Kimbrough harvesting some lettuce from the Veggie chamber. At this point, we only had one Veggie. We actually have two now. It's about the size of a 2-drawer filing cabinet. And it's a very, very simple chamber, probably way more simple than future food production systems will be, but not as complex as our advanced plant habitat, which is really our plant physiology research chamber, which I'm not going to talk as much about today. But both are available on the space station to help us figure out, where we need to go with future crop production systems. And so we did a series of Veggie technology validation tests. The Veggie-01 tests were done between 2014 and 2016. These were the initial tests, and we conducted these with the red lettuce as well as with zinnia flowers. And we had some major milestones from these testing. We got approval for the crew to consume the produce that was grown in Veggie. But we also had some big obstacles. We had a lot of challenges with the watering, and we did get some fungus, and I'll talk about that in a minute. The Veggie-03 series has been conducted since 2016, and it's currently ongoing. We just keep adding letters to our Veggie-03 name. And we've conducted a variety of leafy crop tests as part of this series. We've added repetitive cut and come again harvest technique to this, and we've also looked at mixed cropping in the same chamber. And so if the challenge, the top challenge, which I think is really probably our biggest one right now, is to identify and demonstrate effective options to provide both water and oxygen to the root zone in microgravity. Plants need water, but they don't want to be in a flood. They also need oxygen in the root zone. And because air and water don't mix well without gravity, this is actually really tricky. So when we have insufficient root zone water, as you can see in this image here, we have problems. And here we actually have some plants that died because they ran out of water. This was the very first test of Veggie, and the astronaut intervened pretty quickly, but not quick enough to save the plant that he's pointing out there. So this is, a lesson that we've learned, and we're kind of walking a tightrope, and we fall off a lot with Veggie. We've also had excess water. This was actually during the zinnia grow out, and you can see the condensation on the Veggie bellows there. This actually did not, the result was excess water, which caused stunting and chlorosis of plants and led to some disease issues, which I'll talk about soon. But it actually wasn't because we gave too much water. The problem, at the time we thought that was the issue, but we realized there was a fan malfunction, and the fan had actually been off for quite a long period of time, and water was building up. And as Ray said, the boundary layer gets thicker and thicker in microgravity. And so the water just had no place to go when we didn't have the fan working. And now we've realized that this is a problem that when the hardware has power cut to it and then power is restored, even though the fan says it's back on, it didn't come back on correctly. So it can be manually reset. And now that we know about this, we haven't had this problem. But it's still very easy to overwater the plants. You have surface tension and capillary action kind of ruling when you don't have gravity. And so the water will coat plants, will coat surfaces, and can cause a lot of problems. So what are we doing about this? Well, we have ground-based food production demonstration test beds, water delivery test bed units, to evaluate and compare candidate water solutions. And NASA actually recently awarded 5 grants to different universities and industry partners to develop water and nutrient systems that will effectively provide both water and oxygen to the root zone of plants. We've been testing active and passive concepts, and I think a variety of things has been looked at. And we've done some really interesting work with microgravity fluid physicists. This is the community that's figuring out how to move liquid fuel to engines without gravity, but they're also helping us to figure out how to move water and gases, oxygen, to plant roots. And so with their advice and design ideas, we're testing new concepts in microgravity. We've been working with student engineering design teams. So if you look at the pictures on the bottom, the first one is a concept where porous tubes are embedded in a substrate. This is very similar to what we're using in our advanced plant habitat now, and this is an active system. It works pretty well, but it perhaps is not the most sustainable. We don't want to be launching substrates. So we're looking at variations on this theme with porous tube. The second picture is actually a 3D printed root zone that an engineering design team of students developed, and that was a really interesting concept, and there's a lot of room, I think, for potential there. The third is a traditional ground-based hydroponic system, and we're working with those fluid physicists to see how that type of a system, like a nutrient film technique or a recirculating hydroponic system, can be adapted for spaceflight, just using the properties of fluids and materials and, you know, channels and architecture that keep the water where we want it and inject air bubbles when we want it. So we're really intrigued to see how that will work, and we have some testing that'll go on early next year. And the third concept is one developed at Kennedy Space Center by Howard Levine and colleagues, and then modified by TechShot and Tupperware brands in conjunction with the fluid physics. This is the ponds unit that will be tested in Veggie soon. So we're hoping with all of these different parallel tracks to make some real progress in this important area. The second challenge is to investigate the relationship between microbiomes and food safety. You know, how to effectively sanitize the produce with few consumables and low inputs. We don't want astronauts spending a lot of time and effort trying to clean their vegetables, and we don't want to be shipping a lot of materials into space to do that. So how do we make sure that the produce is safe for them to eat? How can we also control the biological stresses and any pathogens? You know, there are a lot of microorganisms on the space station. There are things inside the seeds. There are things, you know, on surfaces that we send. How do we keep those from being a problem for the plants, plant pathogens, as well as human pathogens? You know, there are a lot of human-associated microorganisms just in, you know, the astronauts and in the vicinity. And so how do we keep those from growing on our plants? And can we use the microbiome to protect the crops and enhance growth? So when we collect data from spaceflight experiments, we'll get culturable microorganism data that looks a little like this, where we look at aerobic plate counts of bacteria and total yeast and molds. You can see that this was from the very first test of Veggie. There was you know, pretty consistent in flight, but tended to be higher levels than in our ground in general. The ground had more variation. We were comparing these to standards for non-thermostabilized food, which aren't appropriate standards for fresh produce, but they were the closest approximate standards we have. We don't have any standards for fresh produce yet. Other experiences were the pathogens. So this was the fungus that we got on the zinnia. It's a mold called Fusarium oxysporum. And when we tried to find the source of this, we really weren't able to. So we think this was already on the space station when the plants were stressed due to that lack of airflow and the buildup of water. This fungus was able to opportunistically colonize the plants. And, you know, that's a problem. This is not something that we want to happen, because it then gets really, really hard to clean out of your spacecraft in the future. So we have to keep this from happening and keep our plants healthy. We do a microbiome analysis, a community analysis on a lot of the plant samples that are returned. And here you can see the community characterization from one of the earlier Veggie flights of lettuce. And interestingly, if you look, these are roots, substrate, and wicks are the first three columns in the flight and the ground, and then plants, shoots are the last column. And you can see that the root zone, the roots, the substrate, which is the baked clay that these plants were grown in, and the wick material all have a very similar microbiome, regardless of whether they're grown in flight and ground. They have pretty similar proportions and a lot of community diversity. The shoot zone has a lot less diversity, and it's very different between flight and ground. So these were really interesting results, and we can start to tease this apart and really understand what's going on in the community, what the contributions are from the seeds versus, you know, the ISS environment. So currently and in the future, we're conducting ground-based microbial food safety and microbiome testing of all our candidate crops, and I'll talk more about our crop candidates in a minute. And we're developing fresh produce food safety standards for space. We're working both with the Human Research Program, Advanced Environmental Health, and Advanced Food Technology groups to help figure out what are the appropriate standards for fresh produce in space. We've had a risk analysis performed, and we're also working with industry. We've been working with the Controlled Environment Agriculture Food Safety Coalition to start to figure out what standards for, you know, the general public on Earth are going to be relevant for our produce. And we also have plans to use on-orbit resources when applicable. So we're figuring out how to do that. How can we sample the plants on orbit to figure out whether or not they're going to be safe to eat? Is this even possible to do without destructively, you know, sampling the plant? Because if there's no plant left to eat afterwards, it's kind of a moot point. And especially using things like non-destructive testing or imaging. So we have a lot of work on robotics, automation, and stress and plant health imaging to help us do this better so that we know before there is actually a problem, that a problem could be developing if the plants are getting unhealthy or if there's some indication of contamination. Storage and handling of seeds to ensure that they are viable, free of contaminants, and long-lived is a really important challenge. You know, on the space station, we can regularly resupply seeds, but when we send a crew to Mars, we're not going to have that option. So we need to make sure that the seeds are going to stay very viable for a long period of time. And so before we fly our seeds for Veggie, we surface sanitize the seeds. And you can see here just an example of some lettuce seeds. We're comparing different sanitization methods, and this one worked pretty well. The unsanitized seeds were obviously not very clean, and the sanitized ones were. But then we also have to make sure that these sanitization methods don't impact germination. And so we do immediate germination tests, but we also do long duration stored germination tests, because some of the seeds that we're sanitizing actually will sit on the space station for years before we have the opportunity to grow them. And for Mars, it's going to be longer. And so we need to figure out if the methods also inhibit the stored germination. And so currently we test each new seed type with different approaches to see what's the best way to sanitize seeds, whether it's chlorine gas, ethanol, other dry approaches. And we've actually started doing a big project on cold plasma to use as a seed sanitization method. Plasma agriculture is a really growing field, so we're exploring that. And we're learning from commercial growers, commercial greenhouse producers of leafy crops, tomatoes, et cetera. What do they do? And from seed producers, how do they take care of their seeds? How do they treat their seeds to keep from having, you know, outbreaks that are economically very devastating? So I think there's a lot that we can learn from industry. We're also testing methods to handle and store seeds with ease in microgravity. And so this is an example of one of the water-soluble food-safe seed films that we've developed at Kennedy Space Center. The seeds are actually embedded in this film. And this is actually on the space station right now. It will be tested in Veggie shortly. So we're looking at this as a way for the astronauts to easily handle seeds, because seeds are really small and hard to handle without gravity. But if you have a piece of film and you have an arrow saying this way down, it makes life a lot easier. But these seed films could also be used to have nutrients or chemicals or even probiotics embedded in them that would surround the seed and help nurture the young plant and keep contamination down. So I think there's a lot of potential for this and similar technologies. For crop production, we have a lot of work going on identifying potential candidate crops that will do very well in our environment. And when I say do very well, I'm looking at things not just yield. Yield is very important, but also the nutrient quality of the crops, the organoleptic attributes. How does that crop taste? And maybe psychosocial benefits. Once we are good at quantifying those, how do the plants make people feel? All of those are really important, and it's hard to define what best is when you are looking at a number of characteristics that aren't always, equally good. So figuring out what's the best approach and how to keep those crops healthy is really important. We assess them under different light recipes, and Ray talked a lot about LED lighting. We can change the spectrum and determine whether or not that will impact the yield, the nutrition, the organoleptic attributes, etc. The microbial food safety may also be impacted by the light. Factors such as the elevated CO2 that we find on the spacecraft, it tends to be, quite a lot higher than it is on Earth, or so in this closed environment where you have a number of people, this elevated CO2 may really impact crop growth. And fertilizer requirements, and how do we adapt those to become more sustainable? So when I look at crops that were tested for veggie, originally we tried a lot of different things in plant pillows, and here are some of the ones that did well. There were a lot that didn't do as well. And we assessed both edible and ornamental crops because, we really wanted a good spectrum. We did grow zinnia specifically to see how flowering and seed formation would work, because that's going to be really important for fruiting crops as well. But we may or may not have ornamentals included in the future system. The astronauts generally want something they can eat, so we're looking for very attractive edible plants. At this point, we do a lot of our crop testing more like this, because there's so much work on water and nutrient delivery, and because the water and nutrient delivery system really impacts how the plants grow. Growing something in a plant pillows may not be representative of what a future hydroponic approach will look like, but we feel that the magnitude of changes will be similar. And so these are a substrate-based system where it's kind of optimized for good fertigation. We can compare plants side by side in this system before testing in more flight-like hydroponic approaches. And so this is sort of our initial crop screening methodology, where we then take samples of the plants, do nutrient analysis, do food safety analysis, and we're able to really quantify. We also send them off for organoleptic analysis, so we can really quantify which plants are good candidates. We grow these under flight-like conditions of temperature, relative humidity, carbon dioxide, and we'll test the LED lighting that these crops might grow well in. When we get samples back from space, we do chemical analysis. We look at elemental concentrations. We look at things like antioxidants and phenolics, and we see, do these vary between flight and ground? In general, there are not that many differences, some slight differences. So, and we're trying to pin down where those differences might be coming in. And we get some feedback from the crew. So this is more in the behavioral health side of things. We've had a lot of anecdotal data from the crew about how much they like eating the fresh produce, how they love the, lobster salad lettuce wraps that Peggy Whitson designed and played. How, Peggy really enjoyed eating and growing the crops. She said she loved growing cabbage, she loves gardening, and it's just as fun in space. She just wants room to plant more. So we've gotten a lot of great anecdotal evidence like this over the years from the astronauts, and now we're actually trying to get quantifiable data in this area. So currently and in the future, we're assessing a wide variety of new crop candidates, both leafy crops, fruiting crops, leguminous crops, herbs. We do preliminary screening with school children. So we have a citizen science program called Growing Beyond Earth in collaboration with the Fairchild Tropical Botanic Garden in Miami, who runs this program. And so far we've had, on average every year, 200, this year I think we're going up to 250 middle schools and high schools around the country helping us to select crops for space. if you have 50 different classrooms growing things under different altitudes, different humidity levels, some with air conditioning, some without, and 50 different groups of children watering plants differently, it's a real robustness index for crops. And crops that do well in all of those different environments might be really good candidates for space. And so far, we've actually had a number of their crops down selected and tested in our environments at Kennedy Space Center, and 2 varieties which have flown in Veggie on the space station. So not only are we getting data from this that's really helping us, because we have a limited amount of space and personnel to do this type of testing, so it helps us to narrow in on really good candidates, but we're also helping the next generation become scientists and get inspired to do space biology. So it's a win-win. We've also had recently, and we have in the future, the Veggie-4 and Veggie-5 experiments, which include the psychosocial and organoleptic evaluations to measure the produce impact. So we actually have the astronauts filling out questionnaires on how plants make them feel and impact their life on space station, and how the plants taste. And we're going to be doing these types of measurements with a number of upcoming experiments in addition to Veggie-4 and Veggie-5, including some chili peppers. So that's pretty exciting. So I'll just end with this kind of showing a range of our folks at Kennedy Space Center and the work that we're engaged in. As I said, in addition to Veggie, we have the Advanced Plant Habitat on orbit, which is the much more high-tech plant physiology research tool. We're learning a lot from both of those, and that's really helping us to inform the future. But as you can see, there are a lot of open questions, and there's a lot of room for work and help in this field. So thank you very much.
Great, thanks for that, Gioia. I think I've got just one question I'd like to ask for both of you, so both of you can try and answer this one. It's really, where do you hope we are in 10 years from now with, say, crop production and plant science in space?
Want to go first, Ray?
Okay. Well, I hope that we have reached the surface of the moon by that time, so we can at least do some very small but exploratory tests in a fractional gravity environment. I think that'll be very informative. I think Gioia touched on an area that is just moving at amazing speed, and that's really understanding the microbiome of plants. And I think 5 or 10 years from now, we'll be a lot better informed on some of those interactions and potential controls that we can implement into plant systems. So I'll let Gioia add into that.
Yeah, I would agree with that. I think to add, I'm hoping, because there's so much horticultural research in universities around the world on LED lighting, I'm hoping there's a good generalized consensus of generalized spectra to use, because it seems like right now every individual plant behaves a little bit differently. And so coming up with kind of a, not one size fits all, but, maybe a few good options for lighting that will do well for all the plants would be, I think, a real relief, because it's still kind of a Wild West. I think we're also going to have a good plan for what we want to do for a future Mars mission and for lunar surface. And, we'll need to be able to start testing that out on the moon before we go to Mars. So I'm hoping we get to the point where we can start testing what a Mars mission will look like on the moon. But yeah, I think, there's a lot of potential in all those areas. The other thing that I think there's some potential in, and we didn't really talk about it today, but I think there's a lot of potential with genetic engineering for crops for space, and custom space crops, essentially. We've done a lot of work to try to make our environments fit the plants, but I think there can be a lot of work for breeding or selective targeted gene modification to get the plants to fit the environment and to help meet some of these needs. And I think that's going to be a really interesting area that's going to take off.
Mm-hmm. And one of the biggest challenges, I guess, to getting to where we're producing crops for on the moon or for a trans-Martian flight?
Well, there's a lot of competing needs for resources. Mm-hmm. And so I think we have to be as efficient as possible and come up with a really good, one of the biggest pluses of plants is also one of the drawbacks in that they can spread or contract depending on the area available, right? And so we're scalable. We're almost infinitely scalable, but I think that also puts us at a disadvantage because when mission are coming with so what do you require? How much space do you need? How much mass, volume, power? And we're like, well, we can fit in wherever we can fit, and so. I think we need a good consensus, a good plan for what makes sense. And I know Ray can talk about there's been some work and, this, like a 1 meter square volume will give you this amount of produce or whatever. 1 meter cubed volume, would give you a certain amount of produce or productivity or salads per crew member or whatever. So I think we just need to plant a flag and say, we need this, and then, work from there because we've been maybe a little too flexible in the past.
Mm-hmm. All right. Thanks, Joy. Do you want to add anything to that, Ray?
Yeah, I think demonstrating sustainability is going to be important too. And so that's going to take the right kinds of experiments and preparatory events on Earth. A baseline Mars mission is nearly 3 years, 6 to 9 months of transit time in each direction, 500 days on the surface. And so, really, we ought to do a 3-year kind of experiment and probably repeat it to have some confidence before we understand the issues of, are the approaches we're doing sustainable, and are the predictions we are making of consumables and what we can recycle from the spacecraft habitat, are those accurate? So I think those will be important tests to do as well.
Mm-hmm. Okay, thanks. Actually, I just want to make one comment, Joy, on and that is, coming from a crop background, I also like to emphasize that breeding is huge, right? If you look at what the crop companies are doing and how deeply they can see into the genomes and how many alleles they're combining to make, single plants for crops, trying to idealize crops is a great goal, but we should also remember that breeding has a huge, it's just an enormous part of that whole thing. And in fact, the crop companies say, if you haven't got a great chassis, there's nothing that you can bolt onto it that's going to make it better. So we really need to make sure that we do have the best underlying genetics to start with in that process, I think. So there's a lot we
Definitely.
can learn in that area too. But.
Yeah, no, definitely. I think it's really a package deal, and figuring out which crops are going to grow well in the environment. And I think work can be done from all directions on that target.
Absolutely, and it's very much a cooperative effort, I think, on that. It's a multifactorial problem, and it's going to take a multifactorial approach to solve it. So.
Yeah, and I think a lot of the work that would go into solving our challenges in space may be very relevant for controlled environment challenges on Earth. Indoor agriculture is something that's becoming, much more prevalent in urban areas around the world. And, as our population continues to grow and farmland does not, I think that's going to play a bigger and bigger role. So work that gets done for space, I think, is also going to be extremely relevant for those environments.
Mm-hmm. I absolutely agree. All right. Well, I'd like to thank you both for your time, and so thanks for doing this, Gioia. Thanks for participating, Ray. It's great that you can do that. So really appreciate it. So I guess we can go ahead and end the recording now.
Thank you, Bruce.
Okay.